All right, we're going to look at the action potential just a moment. And remember, the acetyl-CoA is released in the synaptic cleft, is bound into the acetyl-CoA. All right, we're going to take a look at the action potential, and we're going to remember that acetylcholine is released into the synaptic cleft, bound to the acetylcholine receptor, which allows sodium in the cell and potassium out. So a channel that is gated, the gating is controlled by the binding of the acetylcholine. The flow of ions, both are sodium and potassium, and the direction of the flow is because of the gradient. Now, as it goes down the cell, the longer you, or the farther you get away from that initial end of the cell, there are no other receptors that are the acetylcholine receptors, and what happens is the channels that open up are what are called voltage-gated channels. The voltage-gated channels include the sodium and the potassium, and now the sodium and the potassium channels are individual, and they will allow selectively the sodium to flow in and allow selectively the potassium to flow out. And so even though it is negatively lined where sodium can go in, potassium is not optimized for that particular size. And again, even though the polar region where the channel opens up is negative, sodium cannot get outside or flow through this particular channel because of its size. As we come all the way down the nerve cell, we have lined sodium and potassium all the way through the membrane until we get to the end of the nerve cell. At the end of the nerve cell, there are associated calcium channels. Calcium channels are then activated once it detects the change in the potential or a flow of ions, and calcium channels open, and there's a high calcium concentration on the outside, low calcium channel on the inside. Calcium will flow inside the cell, and what will happen is this calcium will allow the membrane of these vesicles that have acetylcholine inside of them to fuse to the membrane and be released in the cleft. Now, when it's released in the cleft, then we start our cycle over again, but what we're going to learn are there are proteins that are on the surface of the vesicle, and there are proteins on the surface of the end of the nerve, and upon the release or in the presence of calcium, the two proteins will come together and bind tightly. That binding will cause the fusion of the membranes. We'll look at that later in, if we have time, when we go over the article. Now, one thing about the gated channels, what I indicated to you was that in all biological systems, there is a turn on and then a turn off. And in this case, the turn off time scale for the acetylcholine receptor is in the millisecond time scale. And so in the closed, remember, we've twisted the alpha helix right at this very this crux point where I have very large side chains, which then physically closes the hole or the channel. When the acetylcholine binds in two places, both on the alpha subunits, as there are two alpha subunits, we get twisting of the alpha helix, and you get small R groups in this area, and allows the sodium and the potassium to flow. Now, upon this opening, there is a desensitization or an adaptation that occurs in which the channel will rotate back into the closed position even when the acetylcholine is there. This is part of its turn off mechanism, and this allows the acetylcholine, which is bound in the alpha subunits, to dissociate and then eventually bind to the acetylcholine esterase so it can be broken down. Part of the reset mechanism is to use the acetylcholine esterase to break down all the signal molecules, leading to a reset receptor that can be excited again. Now, when we take a look at the insulin receptor, we're going to take a look at the signal of insulin in the body and what it does within the cell. Now, insulin, when you think about it, is associated to sugar and sugar regulation. And what it indicates is a well-fed state, lots of energy resources in the form of glucose in the bloodstream, and as a function of that, what we want to do is to be able to take the insulin and signal to the cells, take in the sugar so that we can either use it or store it. And that's traditionally what you think about when we think of insulin. And if people have can't process insulin well, whether it's not produced into the body or it's not actually bound at the right places, we talk about a disease called diabetes. And so we'll learn about diabetes later, but we're going to talk about what insulin does. For insulin, there is an insulin receptor. Okay, the insulin receptor is a dimer, has an alpha and a beta subunit. The alpha subunit houses the binding pocket for insulin. Insulin is a hormone, but insulin is a hormone. The structure of insulin is actually a small peptide that is released. That peptide is folded in a certain three-dimensional structure, which fits into the pocket of the alpha subunit. The beta subunit becomes a catalytic subunit and has a protein kinase activity. Now, the protein kinase activity, the enzyme that is named kinase, is an enzyme that adds a phosphate. So it adds a phosphate to a hydroxide. There are two classes. There is a class that will phosphorylate serines and threonines, and there's a class that will phosphorylate tyrosines. Both classes have hydroxides, one aliphatic, the serine threonine, one aromatic, the tyrosine. In this case, the insulin receptor will be a tyrosine-specific kinase. All right, what you see in this picture here is that once the insulin binds in this dimer and another one gets together, what happens is the receptors will dimerize. This is the active state. Now, insulin binds, the receptors dimerize. One bind, two dimerize. So they create a dimer. Three, what we get is an autophosphorylation. Now, autophosphorylation would mean I'm going to phosphorylate myself, but what really happens is the dimerization is important. This kinase will phosphorylate a tyrosine onto this kinase. It is essentially phosphorylating a version of itself, not itself, but a version of itself. This tyrosine kinase will phosphorylate correspondingly the other tyrosine kinase. The tyrosine that gets phosphorylated has two different functions. So the third thing after dimerizing is autophosphorylate. Okay, and that gives us two outcomes. One, the first outcome is that the kinase activity goes up. It becomes a better working kinase. Two, creates a binding site for substrate. Okay, and so most proteins will not interact with this tyrosine kinase unless there's a phosphate in this position. If there's a phosphate in this position, then we effectively bind to target. We bind the targets that will be functionalized by this enzyme, and so I now have a tight binding of the substrate to the enzyme. The enzyme, once it's phosphorylated, works faster or better, and then it will phosphorylate the substrate. And so here is the substrate, and here is the, I'm going to use some terminology that we looked, the product. Now, what it does is it converts a targeting protein into a second messenger, a second message. This second message right here is mobile and can move through the system. And so, insulin binds, receptor dimerizes, I phosphorylate, creating a binding pocket that substrates come in, and once it's phosphorylated, it works better, sending out a signaling molecule, a second messenger. Now, if I take a look at this, it's kind of scary. Very long pathway, but we've learned parts of this. Okay, insulin binds, receptor dimerizes, phosphorylate here, phosphorylate there, and here's the substrate. Now I give you a particular substrate, IRS1, that is insulin receptor substrate version 1. If you ever see a version 1, that means we're going to look and find a version 2 or 3 in other places. It effectively binds. This kinase is now more active and it can phosphorylate. Right here, we get amplification because this kinase can phosphorylate many IRSs. Even though we're going to show one, there are many kinases that are phosphorylated. Now, the IRS will then bind to a protein called GARB2, growth factor receptor bound protein, version 2, and then bind to SOS, son of sevenless, which then binds to a protein called RAS. Okay, this is an important protein. We're going to come back to it later in the chapter near the end. Now, RAS stands for rat sarcolemma protein. If you recognize one particular word in this, okay, you have sarcoma. Okay, we all know what a protein is. We know what rat is. Sarcoma references a condition or a disease of cancer. And so it was first identified in the rat, and then we found a homologue in humans, and for some reason we are now calling all of these proteins RAS proteins. Okay, now once these things bind, what happens is each of these binding events causes small conformational changes or shape changes in the protein, and the shape change in the RAS will do two things. One, it will exchange a GDP, a G diphosphate, and it will bind a GTP, a triphosphate, into the protein. First thing, I exchange what's bound. The second thing is, once the GTP is in the RAS, then what's going to happen is we're going to activate what's called RAF1. Now, what's really important, and I put right here, is it's a PK. That's not a Petri kit. Okay, now the formal name is rapidly accelerated sarcoma kinase 1. And so, again, it is a kinase. That's what the important part is. It's a protein kinase. It's an enzyme. It will bind a substrate. The substrate is MEK, mitogen activated protein kinase. Well, that's an interesting name. And this is its inactive form. Once it is phosphorylated by RAF1, it will become phosphorylated and become active. It is a kinase because it is a kinase that's going to phosphorylate MAPK, which is a kinase. That's why it's a kinase. It phosphorylates a kinase. Now, MAP1, mitogen activated protein kinase, will be then in the inactive form to the active form, MAP kinase that is phosphorylated, and this now becomes effective in phosphorylation. The second thing also, once it's phosphorylated, it moves into the nucleus. When it's in the nucleus and phosphorylated, it is active to phosphorylate ELK1, which binds to SRF, and when this combination of the SRF, ELK1, and phosphorylated version binds together, it becomes a transcription factor, which will then interact with DNA, upregulate and synthesize new proteins, which at the end stimulates cell division. Now, we said insulin deals with sugar. And at the end here, I say that it ends up doing cell division. Well, that wasn't initially what you thought about insulin. But if you think about it well, insulin is a signal of the well-fed state. Well-fed state will allow us lots of structural components and energy so that cell division is upregulated. We have the ability to have the energy to do synthesis, and so insulin is a signal for growth. Not the first thing you think about insulin, but I wanted to show it to you. All right. Each time there's a kinase, we have amplification. Each time the kinase is active, and right here, that kinase, we're going to get multiple targets phosphorylated, and I get increases each time I go through this process. And so I might effectively phosphorylate 10 of these. They will upregulate 10 combinations of these. Then I might do 10 of those. 10 times 10 is 100. I might have 10 of these. 10 times 10 is 1,000. And then here I can do 10 of those. So I have 10,000 transcription factors upregulated from the signal of 2 insulin molecules. So we have amplification. Now, I will want you to understand how to turn all of these things off. Well, if you have a biological system, whenever things are turned on, we also have to turn them off. So the first thing that I do in an off state, I have to stop insulin formation. And so I have to get rid of the signal. When that signal goes down, then the dimerization comes apart. When the dimerization comes apart, then I have to dephosphorylate this enzyme to stop it from binding to the IRS and from being active to phosphorylate it. But just stopping that doesn't stop the process because we have tons of IRS that have been phosphorylated. So what I'm going to have to do is take off the phosphates of IRS. Now, in each of these cases, when I take off the phosphate, a kinase puts on a phosphate, a phosphatase will be an enzyme that takes off the phosphates. Once I've done that, I can take it off. Now, here RAS has its own off mechanism. The RAS has what's called a GTPase activity. Now, ACE means breakdown, GTP. It is enzymatic in the fact that once the triphosphate is bound, over time, it will eventually break that down into GDP, which is the off form. If it's not associated to a IRS that's phosphorylated bound to this entire group, then the GTP will stay. It'll be inactive. If that's inactive, then this kinase RAF1 becomes inactive. I've stopped its activity. But if I just stop this activity, I have all of this kinase that's in the phosphorylated state, so I have to take a phosphatase, dephosphorylate that so it doesn't keep upregulating MAP kinase. MAP kinase then has to be dephosphorylated and translated back so that it doesn't phosphorylate ELK1, and finally ELK1 has to be dephosphorylated by a phosphatase so that the CRF and the ELK1 will dissociate and not be a transcription factor. So I can teach you or ask you to describe both an on and an off state, and you have to turn off everything here. One particular big piece of the puzzle will be this RAS and the fact that it has its own off signal as part of its functionality. We'll come back to that at the end of the chapter. Now, did I lie to you that insulin does affect sugar? No, I didn't. Okay, remember, insulin binds, the receptor dimerizes, it autophosphorylates, creates a binding pocket for IRS, the substrate. It gets phosphorylated, and here IRS can now also bind an enzyme called PI3K, all right, which is a PI, or phosphoinositol 3 kinase. What it takes is a lipid, a phosphoinositol that has 2 phosphates on it, and this kinase will put one more phosphate to make the 3 phosphates. Now this lipid now that has one more extra phosphate will float down the membrane and upregulate an enzyme called PKB, protein kinase B. All right, protein kinase B actually has another name called AKT, which is probably more often used now. And if you work with Dr. Casper and her research, they look at the ATK kinase quite often. All right, so this protein kinase B does 2 things. Now, this dotted line means there's a lot of things that happen, but the upregulation of this protein kinase B will upregulate something called GLUT4, glucose transporter version 4. We learned glucose transporter version 1 in a previous chapter. And what this will do is upregulate the glucose transporter so that glucose will be and will flow through. Now this is a transporter, so it will be a passive because glucose is in high concentration. So I've just eaten that Snickers bar, have lots of sugar. It'll go down the concentration gradient, and we will place lots of sugar inside the cell. Now the one thing about the sugar itself is that sugar concentrations inside the cell don't go very high. What we really need to do is we need to have this glucose polymerized into a storage molecule. That storage molecule is called glycogen. The glycogen molecule is how glucose is stored. So if I have lots of glucose, that is great. The signal of the insulin has said, oh, I'll make glucose go in. But then I also need an enzyme to make glycogen, and that is GS, glycogen synthase. And it needs to be active. Okay, so how in the world do I make this active? Well, I'm going to do something very weird. I'm going to take this protein kinase B and I'm going to phosphorylate this glycogen synthase kinase version 3, and in normal cells before glucose gets in the system, this kinase is active, meaning it is going to phosphorylate. And what does it phosphorylate? Glycogen synthase. And when it phosphorylates it, it will turn it off. Now the IRS meant that insulin's there's lots of sugar, I go through this pathway, and what I do is I phosphorylate this regulatory enzyme, this kinase that regulates this enzyme, and I turn it off. And so for me to get glycogen synthase on, I use pKa to turn off the protein that turns off glycogen synthase. So I don't activate it. What I do is I inactivate the thing that inactivates the protein I want active. So it's a different mechanism. Now, one thing that isn't shown is that what I've got to do is I have to upregulate and cause at the same time. I've got to turn off the enzyme that turns it off, and then I've got to put on a phosphatase to make glycogen synthase go up. Once glycogen synthase is active, I can take all the glucose that's being poured into the cell and make them polymerize into glycogen, which is the storage molecule. Okay, and so to turn off these things, I have to dephosphorylate IRS1. I have to convert or dephosphorylate this back into that to turn this off. I then have to, in this case, dephosphorylate this so it converts into this, meaning active. And when that is active, then I allow this process to go to turn off the glycogen synthase. And that is the off mechanism. Now, one thing about regulation that we see is that here's the insulin receptor, here's the pathway I just talked about in terms of protein kinase B being opened up, but over here is the pathway where IRS, GARB2, SOS, RAS, RAF, and all of these others. Okay, some of these proteins didn't seem like they were ones that upregulated things. They just seemed to bind together. Well, quite a number of these proteins that are part of these pathways aren't necessarily places where we get amplification, but what they are scaffolding proteins. Those scaffolding proteins will come together and make sure that the entire pathway, all of those players, are being they're close together, and if they're close together, that means they can respond to these signals much faster. And so a lot of times some proteins are just what are called scaffolding proteins. They bind to the ones that are active, and they create binding pockets so all proteins come together and form a pathway that are bound, and they are bound both in the inactive and the active form so that we can turn them on faster and turn them off faster. In essence, if you take a look at proteins themselves, even an enzyme will have an active site, part of the protein that is functional. So right here, a kinase, here's the tyrosine kinase portion, but it will also have domains or folds in the proteins that will tend to bind to that particular fold in other proteins. And so what we will do is one of these folds may bind to an adapter molecule or a scaffolding molecule that brings multiple enzymes that need to work together in that pathway. And the more of these domains we've identified them, we can actually look at primary sequences, do sequencing analysis, identify, here's the folding domain. This is the sequence. So this protein could possibly interact with this protein and that protein and this protein and this protein. And quite often we find or discover the functions of proteins or where they work in the body by looking at the domains that it has. Many proteins are very large. They have multiple binding domains that will allow several proteins to come together and form organizational pathways. I don't want you to memorize each one of these or the types or the names. I want you to understand that there are binding domains, and you may look at those in particular in other, when you look at details in other pathways, but I just want you to understand there are binding domains and that we're identifying them. I already talked to you about what this accomplishes biologically. We get faster response to signaling. Now, another thing that we need to look at in signaling is we do need to look at what the signal is on the outside of the cell, and then what is created on the inside that sends that signal. Now, the example of the insulin binding to the receptor and then phosphorylating IRS1 becomes what's called a second messenger. That modified molecule will go bind to something else and cause function. A second messenger that occurs, a big class of them, are taking our nucleotides like GTP, a triphosphate, and we will use an enzyme called a cyclase, in this case guanylate cyclase, will create a 3,5' cyclic monomer, monophosphate. This is not an energy molecule. It is not. It's a nucleotide that is very highly modified, and when it gets into the cyclic form, it becomes a second messenger. This is one second messenger. We'll take a look at another triphosphate that will create a cyclic intermediate in just a bit. This particular cyclic GMP will eventually upregulate a protein called protein kinase G, or PKG. We looked at PKB before, we'll look at PKA later. There are a lot of protein kinases in this case. Because it is cyclic GMP, it's going to upregulate a protein kinase version G. Now, there are two classes of the cyclases, and in this, the cyclases here in this class, represented by two examples, specific examples, are bound to a receptor. So this is a receptor enzyme combination. Now, whatever the signal is, a particular factor or molecule will come in and bind to the receptor. They're different shape for different targets. Essentially, the binding will cause conformational changes, and this enzyme will upregulate. It will take the GTP, bring it in, and it will kick out cyclic GMP, which will cause function. The other is a class that is soluble. And so a signal from outside the cell can get to this one, but that signal is interesting. It's the NO gas, nitrous oxide gas, or nitrogen monoxide, if you want to go all the way back to a very formal GChem nomenclature. This gas, which is a very recently discovered signaling process, will actually bind to the enzyme and upregulate it to start creating cyclic GMP from this soluble signaling molecule. NO is nonpolar enough to actually be able to go through the membrane itself. G-protein coupled receptors is probably one of the largest groupings of receptors. They're also called serpentine receptors. It uses a G-protein that is bound or associated to the receptor to stimulate the enzyme adenylate cyclase. Before we saw guanylate cyclase, this is adenylate cyclase. The difference is GTP going to cyclic GMP, ATP going to cyclic AMP. And so when we do this, the receptor is going to upregulate the enzyme adenylate cyclase to create this second messenger, which will then go and upregulate eventually a protein kinase. Cyclic AMP, one of the major regulatory molecules, one of the major second messengers within the cell. The substrates that we're going to look at are epinephrine and glucagon. Now, what you saw before was insulin well-fed state. Glucagon is under starvation state, a hunger state. Epinephrine is a signal that will hijack the system, and it doesn't matter if it's insulin or glucagon, the epinephrine or adrenaline will override the system and cause metabolism to run its own course in terms of creating or upregulating all the pathways that create energy. In terms of metabolism, those three signaling molecules, insulin, epinephrine, and glucagon, are going to be something that we refer back to all the time in terms of how it regulates and how it will affect our pathways, our metabolic pathways. So here you see you see introduced in this chapter all three. The signaling will start with a receptor, and the receptor, remember, always has a component on the outside of the cell and a component on the inside of the cell, and a membrane-bound portion that spans. What we want to do is have a binding pocket created from the extracellular side that will bind a signal, and on the inside we're going to have conformational changes that occur that will cause an event to happen inside the cell. The 7-membrane transmembrane domain is very common, and we know how it all interacts and it's folded. Very common in regulatory events that go through the membrane. The ultimate goal of that binding of the receptor is to activate what's called a G protein, which stimulates adenylate cyclase, which again makes it cyclic AMP from ATP. And so what we really need to do is show you a little bit of what happens. A hormone will come in and will bind to the receptor. The receptor will activate a G protein, which has 3 subunits, an alpha, beta, and a gamma. it will release the beta and gamma regulatory subunit and take the alpha subunit. And what if you saw in that animation, it drops off a GDP diphosphate, it picks up a GTP. It will then go to this enzyme called adenylate cyclase, take ATP, make lots and lots of cyclic AMP. Okay, cyclic AMP concentrations are very low in the cell in an unstimulated version, and once it's stimulated and adenylate cyclase is up and running, cyclic AMP will rise in concentration. Cyclic AMP will bind to the regulatory subunits of a protein called protein kinase A, allowing the catalytic subunits to dissociate. And once those catalytic subunits are dissociated, then they can phosphorylate things and cause function, or what we're going to call cellular response. So let's go over this one more time. A signal will be sent out. In this case, E could be epinephrine. It's a signal molecule. It binds to the receptor. The receptor is interacting with a G protein. There is an alpha subunit, that's like the catalytic portion, and there is the beta and gamma subunit, which is the regulatory portion. The binding of the enzyme causes changes in shape of the receptor, conformational changes, which then cause two things. One, release of the beta gamma regulatory portion, and then two, causes the GDP, the diphosphate, to be exchanged with a GTP. So a GDP falls out and a GTP falls in. Now the alpha subunits are classified in different classifications. In this case, it's going to be a GS, or a G protein alpha subunit that is stimulatory. You can also find them in the versions of GI, which would be an inhibitory, but we aren't looking at that particular one. GS will be stimulatory. Once the GTP is bound into the alpha subunit and it releases the beta gamma, it floats by itself and binds to an enzyme that we're abbreviating AC, but that is adenylate cyclase, which then takes lots and lots of ATP and converts it into lots and lots of cyclic AMP. So the concentration, normally cyclic AMP is low, and it will then bring it to a very high concentration inside the cell. Once the cyclic AMP is very high in concentration, okay, it will bind to the regulatory subunits inside the protein kinase A, the protein that we're going to upregulate. Protein kinase A has 4 domains. It has 2 catalytic. This is the kinase portion that will bind to a protein and phosphorylate it. And it has 2 regulatory subunits. Now the regulatory subunit has a small sequence that is very similar to the sequence that this catalytic subunit is looking to bind. It has the right shape. Catalytic subunits actually do not phosphorylate every protein. They phosphorylate certain proteins at a certain sequence that has a certain shape, and it will bind effectively to that shape. And when it binds to that shape, what will be here is either a threonine or a serine where a phosphate can be added. In this regulatory subunit, it has a sequence, a consensus sequence, a set of amino acids that are shaped that fit perfectly into this catalytic subunit, but do not have a phosphate, or don't have a serine or a threonine. So the catalytic subunit binds to it, thinks it's going to phosphorylate, tries to phosphorylate, but it can't phosphorylate anything, and so it arrests or is downregulated. And it can't release until it phosphorylates. If there's nothing to phosphorylate, then it will be in the inactive form. What will happen is to allow the regulatory subunit and the catalytic subunit to dissociate, the protein kinase A and the regulatory subunits each have 2 binding pockets for cyclic AMP, so a 4 binding total. When cyclic AMP binds into the regulatory subunits, you have small conformational changes, shape changes, which distorts the shape of this sequence and pops it out of the catalytic sites. The regulatory subunit pulls itself out of the catalytic. The catalytic can't let go unless it phosphorylates by itself, but the regulatory subunit can pop itself out. Once this is open, it will find the consensus sequence in the correct shape and bind to that and phosphorylate things. Let me show you a true application of cyclic AMP cascade.
An enzyme cascade is a system for amplifying a biological signal. A very small number of hormone molecules may suffice to trigger the production of millions of enzyme molecules, catalyzing the reaction of tens or hundreds of millions of substrate molecules. For example, a single molecule of the hormone epinephrine, also known as adrenaline, activates a single molecule of adenylate cyclase in a target cell plasma membrane. The activated adenylate cyclase may catalyze the formation of 100 molecules of cyclic AMP from ATP. Each molecule of cyclic AMP can activate a molecule of protein kinase A, and each of those enzyme molecules may catalyze the activation of 100 molecules of phosphorylase kinase, and each of those enzyme molecules may catalyze the activation of 100 molecules of glycogen phosphorylase, and each of those enzyme molecules may catalyze the conversion of 100 molecules of glycogen to glucose 1-phosphate. That's 100 million glycogen molecules for each molecule of epinephrine.
So what you see is we get a very large enhancement or amplification from one binding of epinephrine and the release of the final product. Now, in terms of what you need to know, this is what I taught you up to this point. Epinephrine, or a signal molecule, binds to a receptor. The receptor undergoes conformational change. The conformational change will upregulate a G protein that's associated to it. The beta-gamma regulatory subunit is released. The alpha subunit allows a GDP to fall out, a GTP to fall in, to create an active alpha subunit that can bind to an enzyme called adenylate cyclase. When this G protein alpha subunit that has GTP bound to it binds to adenylate cyclase, this enzyme becomes active. The product is cyclic AMP from the substrate AMP. I make a very high concentration of cyclic AMP. Cyclic AMP will bind to pKa. Remember, the pKa has 4 subunits, 2 catalytic, 2 regulatory. There are 2 binding pockets for cyclic AMP in each regulatory, and once cyclic AMP binds effectively to the regulatory subunits, the regulatory subunits dissociate from the catalytic pKa catalytic subunits. This is now activated pKa. Right here is what I need you to know for this exam. Now, as a specific example, we said at this point in time we're going to say cellular response. Something happens. Here's an example of cellular response. The enzyme phosphorylase B kinase, which is in an active form, is phosphorylated by pKa, making phosphorylase B kinase. Okay, so this is a kinase that will work on phosphorylase. Okay, the phosphorylase that works on is glycogen phosphorylase, which is normally inactive. But once this kinase is active, it phosphorylates this glycogen phosphorylase into an active form. The activity of glycogen phosphorylase will take glycogen molecule, the storage molecule of sugar, where it's just a polymer of sugar, and its enzymatic activity will cleave one sugar off. That one sugar will then be converted into glucose, of which can be used for energy. And so in terms of if it's epinephrine, we create glucose. Glucose is an energy source, and I now have the ability to run away from the lion in the fight or flight pathway. Now, this example of the cellular response, this is a very specific cellular response. We will go back over this and learn this later in a different chapter. This chapter, from epinephrine to pKa's activity, or the upregulation of pKa, is what I want you to understand from this chapter. Now, overall, big picture, there's amplification each time, and by the time you have all this amplification, one signal can generate a very large number. Now, the animation and the slide have different numerical values. The animation was an example. This figure has a little bit more, it follows a little bit more with the numbers that actually happen inside a cell, in this case a muscle cell, and so it is not that you need to know the numerical value. What you need to understand is there's an amplification process as I go. So one signal molecule can produce multiple products that are multiple steps down the pathway, and so a large effect.